Thanks a lot for that introduction. As you see on the slide, I'm officially now associate group leader of the Clevers Group since Hans Clevers is back in the Netherlands, but still doing a lot of the same things. So, as we all know, immunotherapy is quite a hot topic in cancer treatment. And rightfully so, but we also know that a lot of things need to go right before immunotherapy actually works. And one of these things is that you really need a good target. So we would ideally have a target that's really expressed in tumor cells and it's not expressed in normal cells. But how do we find these targets? There are quite some bottlenecks in this. So one thing you could do is use tumor material to try to find these targets. But this can be difficult because we usually have really limited material, especially if you have a biopsy sample, then there's really not enough tissue to do a lot of experiments with them. And if you're looking at experiments like immunopeptidomics, for example, you really need millions and millions of cells. So that's quite difficult with tumor material. Another issue is that with quite some tumors, you can really have a lot of normal tissue in between. So that can confound your results, because then you're not really sure how many tumor cells you actually have in the sample. So it can be difficult to really know what's tumor specific. Also, once you've done experiments with tumor material, it's quite difficult to really do follow-up experiments in those same cells to do functional validation, for example. You can also use cell lines, which is being done a lot, and it solves quite some of the problems that I mentioned before, because you can keep expanding these cells, you can get enough cells for experiments, so that can be very useful. But the problem with cell lines is that you're often culturing a very monoclonal type of tumor. So this doesn't really capture the heterogeneity that we see in a tumor. So basically, it's not a really good representation of the cancer that you're trying to study. And of course, I'm going to talk about organoids, because maybe organoids would be a solution for some of these problems. First, a little bit of background about organoids. Being from the Clevers lab, I, of course, talk mainly about patient-derived organoids. And these type of organoids were first described in 2009 by the Clevers lab. Back then, Toshiro Sato found that if you isolate intestinal stem cells from the mouse, and you put them in a dish, and you provide them with the right cocktail of growth factors, but also extracellular matrix, that these cells don't only expand and make other stem cells, but they are actually able by themselves to really form self-organized three-dimensional structures that don't only contain of stem cells, but also of a lot of different functional cell types that can be found in the intestine. So that's a very useful model to study things that are actually happening in these cells. Then later, they found that it's not only possible to do this from intestinal cells, but also a lot of other organs. And it's not only possible to do it from mouse tissues, but also for humans and other species. And it's also possible to do this from cancer tissues. So this can be a very useful way to really expand your tumor material in a way that you're still preserving some of the tumor cell heterogeneity that we see in vivo. Because we've also seen that these models can really recapitulate some of the key characteristics of the tumor that they're derived from. So in 2015, Mark van Wetering and colleagues published a kind of benchmark paper where they showed that they could create a biobank of living tumor organoids from colorectal cancer patients. And these organoids could be used for really high throughput experiments, such as drug screens, but they can also be frozen down and then later thawed and started up again for future experiments. So this is a very useful way to use this tumor tissue in research. And when the Princess Máxima Center started, of course, we also wanted to grow these tumor organoids for pediatric cancers. And initially this was thought to be quite straightforward because we just do the same thing as for adults. But we found out that it was a little bit more difficult than that. And that is because many of the adult tumors are actually carcinomas. So they are epithelial tissues. And the original organoid culture protocol was also based on epithelial tissues. So the kind of growth factors and matrix that they need could be easily translated to adult cancers. For pediatric cancers, this is a bit different because we have a lot of sarcomas, we have a lot of neural tumors, and we now know that these can really need a different type of growth conditions. And we also know that we need to optimize these growth conditions for every individual tumor type. So a lot of groups in the Máxima have been working on that in the last years, and we have been able to grow quite some different types of pediatric cancer organoid models. And you can see some examples on the slide. So that's basically the concept. We get tumor material, either a biopsy or a resection. We put this into culture, we grow organoids, we can biobank them, we freeze them viably so that we can start them up again later. We really fully characterize them, and this is very important because we really need to know, are we growing cells from the right patient, but also are we growing tumor cells? So this is an important step in the whole process, and then once you have these characterized models, there are a lot of experiments you can do with them, and these can be more fundamental type of experiments, but also more translational things, such as doing drug screens. And one of the things you can also use them for is to see if you can find antigens, targets for immunotherapy. And of course, very shortly, a little bit about the Central Máxima Organoid Biobank, which is also part of the organoid platform in Oncode Accelerator. Here we really try to standardize many of these steps so that we really know exactly what we're doing. We can write very specific protocols that a lot of people are able to follow. We fully characterize the models, and we want to make these models available for the wider research community. So then about the Demonstrator Project, because of course this session is about Demonstrator Projects, to really show what we are now using these organoid models for. And the title of this project is Oncofusion-driven neoantigens as immunotherapy targets in sarcoma. And this is a collaboration between the therapeutic vaccines workstream and the organoids platform. So this is the team of this project. Sebastian van Hees is the Demonstrator Project lead owner, also lead of Work Package 2. We have Jarno Drost, who's the lead of Work Package 1, also from the Máxima. Fabio Marino is a partner in Work Package 2 and involved in immunopeptidomics. He's from the Utrecht University and also getting more involved in the Máxima. Sjoerd van der Burg is lead of Work Package 3 from the LUMC. Simon Venneman is program manager who is working a lot for this project and also helped us a lot in getting it. Albert Heck, also Work Package 2 partner from Utrecht University. And myself, I'm a partner in Work Package 1 and I'm the study director of this project. But these are maybe the least important people because we have a lot of other people involved who are actually doing the work for this project. So these are actually helping us, or well, really doing the work to actually get some results. So the goal of our project is to identify and validate antigens that can be found in Ewing sarcoma and in fusion-positive rhabdosarcoma. I will tell a little bit more about what these tumor types are. And we do this by leveraging organoid technology, but also multi-omic approaches. And we hope that eventually that the antigens that we find will in the future serve as the foundation to develop cancer vaccines. So about the tumor types that we're studying, both are in the pediatric setting. Ewing sarcoma is mainly a bone tumor, and it's a type of sarcoma that's always driven by a fusion gene. And in most cases, this is a fusion between Ewing sarcoma region 1 that is fused to FLY1. And this type of fusion gene can cause a lot of aberrant transcription, a lot of methylation effects. So this will have a very important impact on those cells. This is mainly a tumor that occurs in teenagers, young adults. And we have rhabdomyosarcoma, which is a muscle-type cell tumor. And there are actually several subtypes of this, but we are focusing in this project on the alveolar subtype, which is also driven by a fusion gene. And in this case, it can be either PAX3 FOXO1 or PAX7 FOXO1. And it's also mainly in teenagers. For both of these tumor types, in the Maxima, we have been able to grow tumor organoids. So we've developed the protocols that we need to do this, and we have the lines at the Maxima in place to do more experiments with them. So what does our demonstrator project look like? In work package 1, we are mainly working with the organoids. So we are developing and also validating interferon gamma stimulation protocols because we know that this can help the tumors to express peptides on HLA molecules. We want to select lines that have HLA types that are quite common in the general population. And we then also analyze actual protein expression with flow cytometry. Because we will need a lot of cells for the follow-up experiments, we are really scaling up the cultures to get millions and millions of cells. And as before, we also really want to characterize these cultures to know what we're really dealing with. Then in work package 2, we are using a lot of techniques that are present in the therapeutic vaccines work streams. First, we're going to do RNA sequencing, both short read and long read, also to look at the data itself, but also to really have a new map, a new transcriptome that we can build to map the ribosome sequencing data to. Because we will also do ribosome sequencing to really look at which RNAs are being actively translated in these cells. And in addition, we will also do immunopeptidomics. So we want to see which antigens are actually being presented by HLA on the cell surface. And putting this all together, we hope to get a top list of potential neoantigens that we will then use in work package 3. And basically in work package 3, we want to see whether the antigens that we have detected are really able to generate an immunogenic response. This is the Gantt chart of our project, and you don't need to look at all the letters, but it's just to give a little bit of an overview that at the moment we're still in the first phases of the project. So we are quite far with work package 1. We're doing a lot of the interferon gamma testing. We have already done a lot of FACS experiments and done a lot of scaling up, so we're quite close to finishing work package 1. But then, of course, the actual interesting part of seeing what data comes out will start. So hopefully in the future we will have a lot more to tell. And then a big thank you to everyone involved in this project. Again, the same team on this slide. Collaboration between Princess Máxima Center, Utrecht University, and LUMC. And of course, a big thank you to Oncode Accelerator for the funding and for this invitation. And Carlijn Wilschut is helping us a lot in the project management and everything surrounding it. And thanks for your attention.